Thank you all for coming. So, my pleasure to introduce Sophie Lee today, who will be talking about her thesis. So when Sophie came to see us, or see Andrew a few years ago, full of enthusiasm for undertaking some basic research, he didn't hesitate in terms of getting her into the lab for a PhD. And that enthusiasm has been pretty much unwavering throughout her entire period with us. She's an extraordinarily affable and very approachable and friendly person in our lab. It's been a pleasure to have. Now, in terms of her thesis throughout this period of time, she's been looking at a fairly interesting region of imprinting, where several genes have been implicated in stem cell and leukemia, as well as angiogenesis, and is, I think, a testimony to her development as a student as well as a critical scientist, that her persistence and perseverance through logistic and somewhat technical challenges has actually been brought to fruition in the last year or so. So I think you'll be very interested to hear what Sophie has to present today. I know Andrew, myself and Lee and Warren have been very impressed with her approach throughout the PhD, and so I'd like to welcome Sophie.
Thank you. Thank you, Ashley, for that kind introduction. Thank you, everybody, for coming to my PhD completion seminar today. So today, I'm going to talk about my PhD project, which was the role of Dlk1 in haematopoiesis, leukemia, and angiogenesis. So this is a brief overview. Dlk1 is not familiar to a lot of people here, so I'll just give you a very brief summary. And then main data will be, I will discuss it in 3 sections. The first one on the role of Dlk1 in stem cells and haematopoiesis, the second section on Dlk1 in leukemia, and then in angiogenesis. So Dlk1 is a short for Dlk1 homolog. It's a transmembrane and secreted protein with 4 major isoforms due to differential splicing. So here is a schematic diagram of the Dlk1 protein. Signal protein followed by the EGF-like repeats, which makes it a member of the epidermal growth factor family, followed by the protease target region and transmembrane region and intracellular tail. So the protease target region here is the important bit where the ADAM17 protease acts to cleave the protein here, and this bit then becomes a soluble or secreted form, and this part is then left behind. So the mechanism how multiple isoforms can result is due to differential splicing of exon 5, which is the biggest exon of Dlk1 and also the last exon. So in the first isoform, which is the complete form or the long isoform, where the gray box here represents, when translated, the protease target region. Obviously, this form can give rise to secreted forms, and so does Dlk1b, which, despite the splicing of a lot of exon 5, still possesses this protease target region and therefore still can give rise to secreted form. But these 2 forms here, which is can only exist as membrane-bound forms because the splicing of the protease target region means that the protease cannot act, and therefore they can only exist as membrane-bound forms. Now, it's quite an interesting protein. It's been widely published as to be a non-canonical atypical Notch ligand. There's some controversies about this, and some of its actions are not due to not have anything to do with the Notch signaling, but most of the literature supports that it is an atypical Notch ligand that works as an inhibitory factor. So another really interesting characteristic is that it's an imprinted gene. So imprinting is quite rare. It's an epigenetic phenomenon and allows for parental-specific gene expression in diploid cells. In Dlk1, it allows for paternal monoallelic expression, and the maternal allele is silenced. And it belongs to this intriguing region called Dlk1 DO3 imprinted cluster. And it's one of the 3 paternally expressed genes, the Dlk1 DO3 and RTL1 paternally expressed genes in this cluster, and the Dlk1 and DO3 seem to be sort of ancestral genes because they're preserved throughout the vertebral genomes. All three paternally expressed genes are involved in growth regulation, and quite recently it seems to be the activated Dlk1 DO3 region seems to be a marker of pluripotency and induced pluripotent stem cells, and also not just the coding genes, but the maternal non-coding RNAs also seem to be very important in maintaining long-term hematopoietic stem cell function. So other functions of Dlk1 from the gene knockout and transgenic studies include embryonic growth regulation, adipogenesis and skeletal development, postnatal neural stem cell regulation, and also in haematopoiesis, which is what we're going to talk about now. So the first part of my PhD is, the biggest part, is about the role of Dlk1 in stem cells and haematopoiesis. Dlk1 gene expression has been shown to be enriched in the hematopoietic stem cell compartments, and active expression of the Dlk1 DO3 cluster, as I've said, including or especially the non-coding RNA, seem to be important in the embryonic stem cells. And previous publications have shown that Dlk1 knockout seems to lead to increased number of hematopoietic progenitors. So this knockout Dlk1 phenotype has been published in the past in 2002. As you can see here in the first panel, the knockout pup is obviously much smaller than the wild-type pup, and the BALB/c knockout mice seem to have this particular eyelid abnormality called blepharophimosis, and they suffer from perinatal mortality as well as increased adipose tissue and skeletal malformations and increased serum lipid metabolites. Now, this one is actually only, this one is created from only deleting the exon 2 and 3 of the Dlk1 gene. And as I've mentioned before, the biggest exon is actually exon 5 and 4. So we've actually generated our own Dlk1 knockout mice. And here the entire Dlk1 gene is knocked out. And what we hope to do, which was this part here, we hope to make heterozygous Dlk1 a male and female and get homozygous knockouts and continue to breed the homozygous knockouts to get enough mice to study. But unfortunately, what happened is that these homozygous knockout mice proved to be incredibly difficult to get. Like after 3 years of breeding with very active breeding pairs and lots of pairs, I only got 4 Dlk1 knockout mice and 2 females, 2 males. And when you breed them together, they're completely infertile. They cannot reproduce at all. So obviously this is different from these mice, which are, I mean, they do produce smaller litter sizes, but they can reproduce, and they're not that difficult to get. So for fetal livers, I was able to get homozygous knockout fetal livers to study the fetal stem cells, because they're not fatal in utero. But for adult mice, I just could not get these at all. So we thought of doing this, which actually proved to be very fruitful. So because Dlk1 is an imprinted gene, you can get a knockout mice by another way, which is breeding the wild type with heterozygous Dlk1 male. So it has one knockout allele, and when you get the offspring from this mating, you know if the offspring has a knockout allele by genotyping, that it's come from the father, not from the mother, because the mother is a wild type. So because it's an imprinted gene, the maternal copy is silenced, and the knockout paternal allele is the only one that's expressed. So you really should be getting, the imprinted knockout genotype should give you a homozygous knockout phenotype. Now, in reality, they're slightly different in that they are not as hard to get as the knockouts, and they can reproduce, but they do suffer from the same perinatal mortality and growth retardation, just like these ones here. So I've had to use these mice to look at the adult bone marrow. So it was very difficult. It was very important to make sure that they are true knockouts, at least in the bone marrow. And in a series of validation studies using qPCR and Western blot and flow cytometry, I've shown that the Dlk1 imprinted knockout in the bone marrow, there is no detectable Dlk1 expression. So it is okay to use them to study the adult bone marrow. So I'm not going to show you all that validation study here in the interest of time. So using these mice to look at adult bone marrow and using these homozygous knockout for fetal livers, I wanted to ask the question of, does knocking out Dlk1 have any effect at all in the haematopoiesis or in the stem cell function? So for both fetal and adult haematopoiesis, I've used quite similar experimental approaches. First, looking at the expression pattern of Dlk1, and then looking at flow cytometry data, comparing Dlk1 knockout fetal livers versus wild type, particularly looking at the lineage negative, c-KIT positive, SCA1 positive cells, stem cells, and the progenitors, which are the SCA1 negative cells. And also looking at the clonogicity of Dlk1 negative fetal livers by looking at the colony assays using semi-solid agar, so looking at the colony numbers, and particularly wanted to look at the hematopoietic stem cell, and to do that, I performed serial competitive transplant using homozygous knockout donors versus wild-type donor. And very similar approaches in the adult hematopoiesis, but using, as I've said before, the imprinted knockout mice instead of the homozygous knockout. So first of all, looking at the fetal hematopoiesis, I've used a lot of flow cytometry, so I'm just going to show you an example of gating strategy. So this is it's either fetal liver or bone marrow, where you first of all use the forward or side scatter to gate on the cells of interest, and then obviously you want to select the live cells. And then if you're wanting to look at the stem cells and the progenitors, you want to gate on the lineage negative, c-KIT positive population, and then split it by c-KIT and the SCA1 expression, with the SCA1 positive and c-KIT positive population being the LSK stem cells, and the other one, the progenitors. If you want to look at the mature cells, you gate on the viable cells, and then look at the B cells, which express B220. These are mature cells, obviously. The T cells, CD4 and CD8 negative, or CD4 or CD8 negative. And granular or monocytes, which express GR1 or MEC1. So you can use this gating strategy not only for bone marrow fetal livers, etc., but also for eye bleed as well, to look at mature cells. So by flow cytometry, Dlk1 is significantly expressed in the hematopoietic stem cells using a validated Dlk1 antibody. Now, I won't show you the validation study, but I've validated this Dlk1 antibody against the mouse Dlk1 using, to make sure that it's sensitive and it's specific. So using that antibody, at E13.5 and E12.5, the fetal livers express Dlk1 at significantly high level compared to the negative control. I've used Dlk1 knockout livers as negative control, so both stem cells and the progenitors at both time points. And using a different method, which is using Dlk1 EGFP transgenic mice, where the GFP protein expression is linked to the Dlk1 transcription, the Dlk1 expression, again, is significantly higher in LSK stem cells, and they're actually higher than the mature lineage-positive cells. So here, the transgenic mice is represented by the green line, and the wild type by the blue line. And obviously in the wild type, there's no change through LSK or progenitor or lineage positive cells. But in the transgenic mice, you can see there's a significant shift in fluorescence in the stem cells. And as the cells become more mature, the expression drops. So this is expressed here again in this lower panel, both E13.5, E15.5. There's significantly more expression of Dlk1 in the stem cells compared to the mature cells. And the other thing to note is that in only 2 days, the expression of Dlk1 has dropped quite a bit, and this is also significant. It can also be detected by Western blot. So in the wild-type fetal liver, this is where the Dlk1 sits with all the isoforms. You can see the Dlk1 protein expressed, but not in the homozygous knockout Dlk1 fetal livers. So using the Dlk1 homozygous knockout fetal livers and the wild-type livers, I've performed the agar culture feature agar culture assays to see whether the Dlk1 knockout has any effect on the clonogenicity. And as previously shown in previous publications, the Dlk1 knockout fetal livers appears to lead to a higher number of colonies than the wild type in terms of total number of colonies, blasts, and megakaryocytes. But when you look at when you look at the flow cytometry data, actually looking at the numbers of the LSK or progenitor cells per 100,000 cells, there is no difference between the wild type and the Dlk1 knockout. So, as I've said, I really wanted to look at the function of Dlk1 or the role of Dlk1 in the stem cells. And to look at this, I've done a fetal liver competitive serial transplant assay, which is a little bit complicated. So I'll just take you through it. So here on the left is the 45.2 positive donors. So in all of my experiments throughout my PhD project, all the donors are 45.2 positive and recipients are 45.1 positive. So the 45.2 donors is one, a Dlk1 heterozygous female plugged by the heterozygous male. And the embryos are E13.5. And the competitor is a wild-type female plugged by a GFP male, and that it provides a transgenic competitor fetal livers. So from the heterozygous female, we get the wild-type fetal livers, which are pulled together, and we also get the knockout fetal livers, which are pulled together, and they're put, 2 million of these are put in one tube, 2 million in the other tube, and the heterozygotes are all discarded. And then the GFP competitor fetal liver is mixed with the knockout or the wild-type fetal liver tube in 1-to-1 ratio, so 50-50. And after that, this, the Dlk1 knockout and competitor fetal liver cells are injected to the knockout arm of recipients, so they are all 45.1 positive, lethally irradiated, and 1 million of fetal liver cells are injected per recipient, and the other arm is the wild-type and competitor fetal liver cells in 50-50 mix. And I've done 4 independent pairs of knockout and fetal liver donors. The next thing is the serial transplant, when at 8-week points, you pull all the bone marrow together from the 4 recipients who receive the same bone marrow, and then you inject 2 million of bone marrow cells to the next lot of recipients, which are all, again, 45.1 positive and lethally irradiated. So the main analysis I performed here is chimerism analysis to see the donor chimerism in the reconstituted recipients. And this is the analysis strategy that I've used, so it's quite easy. You can just use eye bleed if you want, but I've also analyzed the bone marrow as well. So this is what the peripheral blood looks like after lysing, and you gauge on the nucleated cells which express the CD45 antigen. And after selecting the viable cells, you're interested in the 45.2 positive cells, the donor origin cells, And from that, you can split that into FITC positive or FITC negative cells. So obviously the FITC positive cells are from the competitor, and the FITC negative cells are from the test donor, either the wild type or the knockout donor. So this is how I assess the chimerism. And you can also look at all the other subclasses of the hematopoietic cells from the 45.2 positive pool, like the granulocytes and B cells and T cells. So using this chimerism analysis for primary, secondary, tertiary to quaternary recipients of the serial transplant, after all of that, there was no difference between the fetal liver cells from Dlk1 knockout donor versus wild type donor in their ability to reconstitute recipient mice. So they were equivalent in their ability to do so. Now, when we look at the adult bone marrow, it's quite different from the fetal liver, which is what I'm going to talk about now. So in an adult bone marrow, you can't actually detect Dlk1 very easily by using flow cytometry or transgenic mice or by Western blot. You can detect it readily by qPCR. So as I mentioned before, the imprinted knockout mice has no detectable expression by PCR, undetectable. So I've looked at the Dlk1 imprinted knockout versus wild type in terms of their peripheral blood, and there is no difference between the knockout and the wild type mice. But when you look at the agar culture assay, again, as we've seen with the fetal livers, the knocking out Dlk1 seems to lead to increased colony numbers in the total number of colonies as well as in the megakaryocyte colonies. And this difference, however, seems to disappear in the older age cohorts. When you use older mice, there's no difference. By flow cytometry, again, the number of stem cells and progenitors, and also the biopotential erythroid megakaryocyte progenitors were examined, and either in the number of cells per femur or in the number of cells per 100,000 viable cells, there's no difference between the knockout and wild type in terms of the number of progenitors or LSK stem cells or in the erythroid megakaryocyte progenitors in the young or in the old age cohorts. So, as I've done in the other fetal liver experiments, I've done competitive serial transplantation assays, so very similar to what I've done with the fetal livers, but obviously using adult bone marrow. Again, 50-50 mix with the Dlk1 imprinted knockout bone marrow and the GFP competitor bone marrow, and now 2 million bone marrow cells per recipient of the irradiated CD45.1 positive recipients. And I've done 3 independent pairs of Dlk1 imprinted versus wild type donors. Now, this was also taken to quaternary recipients by serial transplant. So when I've done that, it was different from the fetal livers in that the contribution from the Dlk1 imprinted knockout donor was significantly lower compared to the wild type donor, especially in tertiary and quaternary recipients with a statistically significant value. So this was quite surprising. So just to tell you the summary before we move on to the next section, Dlk1 is highly expressed in fetal livers, and especially in the stem cells, and their level of expression decreases with age. It's still detectable in the adult bone marrow, but at a lower level. The Dlk1 imprinted knockout mice are smaller in size, and they suffer from increased perinatal mortality. But otherwise, the bone marrow and the peripheral blood analysis do not show much significant difference between the imprinted knockout and wild-type mice. The agar culture assays from both fetal livers and adult bone marrow show increased clonogenicity with Dlk1 knockout, which has been shown before. But the in vivo competitive serial transplant has not been done before, and it shows that the fetal liver did not show any difference to the wild type, but the same assay using the adult imprinted knockout marrow showed that the imprinted knockout marrow was significantly inferior to wild type bone marrow in reconstituting recipient mice, indicative of stem cell defect. So far we've talked about what Dlk1 knocking out the Dlk1 does to haematopoiesis and stem cells. And the next section is going to be about what overexpressing Dlk1 does in leukemia. So in the previous publications, many of them have shown how frequently overexpressed it is in hematopoietic malignancies. So especially this is human, myelodysplastic syndromes, acute myeloid leukemia, M6, which is erythroleukemia, and M7, which is megakaryocytic leukemia, and myelofibrosis and essential thrombocythemia, which are myeloproliferative disorders. And in this study, they've shown that, well, they've studied primary patient samples of leukemia, AMLs, and they found that 76% of AML overexpressing Dlk1 did so due to loss of imprinting, leading to biallelic expression, which then led to the increased Dlk1 expression. And this was due to hypermethylation. Now, another interesting thing that they showed in their study was that in their primary AML patient samples, more than 60% actually overexpressed Dlk1. So obviously it was very frequently overexpressed in human leukemia. And not just in the hematopoietic malignancies, but in the non-hematopoietic malignancies, especially the hepatocellular carcinoma and neuroblastoma, they were frequently overexpressed, and they seem to have a cancer stem cell-like quality with kind of increased collagenicity and self-renewal and chemoresistance. And more recently, the microRNAs from the Dlk1, DO3 cluster region is emerging as kind of new biomarkers of solid tumors. So the questions that were relevant to me was, what was the significance of Dlk1 overexpression in leukemia? Does it actually lead to leukemia? Does it cause it? Or is it just a consequence? Is it a leukemia stem cell marker, like in some other solid cancer cell lines? And can it be a therapeutic target? This is also an important question about prognosis, but I've not examined it in the scope of my project. The other important question is that, does it actually have any impact on hematopoiesis? So to address these questions, I first of all wanted to confirm that I can reliably detect Dlk1 overexpressions in cell lines, and it is actually quite readily detected by both qPCR and Western blot. And in murine cell lines, which is the upper panel here, the ones that overexpressed Dlk1 were mainly the TALL cell lines from the LMO transgenic mice, and also the BALL from immune-MYC transgenic mouse. In humans, megakaryocytic cell line like CMK, erythroleukemia cell lines such as HEL and K562, and HUS7, which is the hepatocellular carcinoma line. And by Western blot, D here is the MCF7 breast cancer cell line, which had no expression of Dlk1, and you can see by Western blot there's absence of expression as well by Western blot. But the other positive cell lines readily show you can detect it on Western blot. So I can detect it, but can I detect it on flow cytometry by an antibody? Because if I can sort the positive from the negative cells and see if the positive cells are any different from the negative cells. So I've purchased two antibodies, the EpCAM and RND antibody. One is directed against this part, and the other against the complete part of the complete human Dlk1 protein. And this is sort of a typical plots that you get with the CMK and the K560 cell line using these antibodies. So gating on these cells and then the live cells, you hardly get any dead cells with the leukemia cell lines. So all of these, and then Dlk1 antibody quite nicely shows this population here for CMK cell line. And here you can see, compared to the isotype control for primary antibody and isotype control for secondary antibody, the red line represented by the Dlk1 antibody shows quite a nice discrete population of Dlk1 positive cells. For K562, it's not as nice. It's more of a spread, but still compared to the isotype control antibodies, there is clearly a positive population there. So while I've used both antibodies to conduct experiments looking at Dlk1 positive and negative cells, and in the meantime, I wanted to do validation to make sure they do what they're supposed to do. And this was quite intriguing in that with the EpCAM antibody, the K562 cell lines that were sorted for Dlk1 positive expression was slightly higher in Dlk1 expression than the negative cells, but the CMK cell line, for some reason, the supposedly Dlk1 negative population had much higher expression of Dlk1 than the positive cells. So it's kind of doing the opposite of what it's supposed to do. But when you look at the RND antibody, this is quite nicely consistent with what it's supposed to do, where the positive cells are higher in expression much higher in expression than the negative sorted cells, for both cell lines. So this is really strange. So I had to repeat it 4 times, just make sure that I'm not mixing something up. But it was every time it was the same. So in the end, I don't know why, but I had to abandon this antibody and just use this antibody from then on to do my experiments. But it was a very strange thing. And this is a Western blot showing the same result as what I've shown by qPCR. So using the RND antibody, when I look at the Dlk1 positive versus Dlk1 negative cells in terms of the agar cultures, growing them for 7 days at 37 degrees, I found that there was no difference between the Dlk1 positive and Dlk1 negative cells in terms of their ability to give rise to the colonies. So this is different from the solid cancer, solid tumor cell lines where they've shown there was more clonogenic. But in my haematopoietic leukemia cell lines, there was no difference. So the next thing I've done is to see whether I can overexpress Dlk1 in vivo in a mouse and whether that causes increase in leukemia or poor survival. So first of all, we have to clone Dlk1, and I've done that by getting the cDNA from ovaries and testes, which is an organ which still expresses Dlk1 at high level in an adult. So after harvesting all the RNA and then making cDNA, I've PCR'd it with primers against the beginning and the end of Dlk1 gene, which is tagged with the BAGL2 and ZO1 restriction enzymes. So this biggest form here represents the complete cDNA, or the long isoform, which I've just named cDlk1 just for the ease of using it. And the shortest one here is the membrane-bound isoform, which I've named MDLK1. So once these were cloned, those genes were inserted into the PMIC vector separately. And we've chosen the PMIC vector because it allows for high-level constitutive overexpression in embryonic stem cells and hematopoietic stem cells because of their unique 5' LTR, which is why we chose PMIC vector, but also because it's got this convenient GFP cassette, so you can tell which cells are transduced or not. So this is why we use that vector. And what I've done is use fetal liver cells from CD45.2 embryos at E13.5. So again, all the donors are 45.2 and all the recipients are 45.1. And transduce them using the retroviruses containing the overexpression vector or the empty control MIG vector. And I've done 5 cohorts of cDLK1 and 5 cohorts of MDLK1, and each cohort was 24 mice, with half getting the overexpression vector and half getting the MIG vector. So I've looked at these recipient mice at 8 weeks and 24 weeks, looking at the peripheral blood count and the facts for the donor chimerism. And also, very interested in whether they develop any leukemia or any hematological abnormalities or any other signs of illness. First of all, I wanted to make sure it does work. So short term, at 7 days, I've first transduced them and then grown it for 7 days and showed that the Dlk1 overexpression vector transduced fetal liver cells do express Dlk1 at significantly higher level than in the MIG transduced cells, which is also seen in the Western blot. So number 3 is the overexpression vector transduced fetal liver cells, and obviously that is the only one that expresses Dlk1 at high level, not number 2, which is the one without any virus, and 1 which is the one with only the empty MIG vector. So using this overexpression vector, first I've looked at the peripheral blood count at short and long term, and it does not affect the blood count at 8 weeks or 6 months, the complete Dlk1 group. And the membrane-bound Dlk1 group, same, with no difference between the control group and the Dlk1 overexpression group in the peripheral blood count, at short term or long term. But what I was really interested in was whether does it cause leukemia was the main thing I wanted to see, and whether it causes poor survival. And unfortunately for me, there's no difference between the overexpression arm versus control in terms of their survival. They're the same. And also in terms of acute leukemia incidence and GFP positive leukemia, there's no difference. It's exactly the same. So when you see that, you kind of think, is it because it really doesn't cause leukemia, or is it because the vector just doesn't work at long term? So do you want to make sure that you can demonstrate that it still works? And at 18 months, this is the bone marrow taken from the recipient mice at 18 months, and just looking at the 45.2 positive and then the FITC positive cells. In the MIG arm, there's no Dlk1 expression as detected by the Dlk1 antibody, the murine antibody. And in the Dlk1 MIG arm, there's clearly a population that expresses Dlk1. And that's just the same data. A is for the cDlk1 group, and B is the mDlk1 group. And again, against the versus the MIG control group, there's significantly higher level of Dlk1 expression in the overexpression vector groups. And also by Western blot, on lane 3 and 4 are the overexpression vector bone marrow. On lane 3 and 4, and 5, 6, and 7 is the bone marrow taken from the control group where there's no expression of Dlk1. So it doesn't cause leukemia, it doesn't cause poorer survival. Does it have any other effect on the haematopoiesis? And one way to look at that is to look at chimerism in the recipient mice. So this is the chimerism analysis at 6 months in the recipient mice, comparing the MIG group versus the Dlk1 group, which is the dark square here. So within the CD45.2 positive cells, or the subclasses of the CD45.2 positive cells, when you compare the proportion of GFP positive cells, there's no difference between the MIG group and the Dlk1 group. But when you take into account the initial transduction rate, then there seems to be a little bit of difference in that. What I mean by that is I always measured the transduction rate just before I injected the fetal liver cells to the recipient mice. So just before, I would take an aliquot and run it on flow cytometry and just measure what the transduction rate was of those fetal livers. And as you probably will know, when you have a vector that's got a large gene inserted to it, the transduction rate is usually, the efficiency is usually not as good as an empty MIG vector. So for me, for each cohort that I've done, the MIG transduction rate was always higher than for the Dlk1 vector transduction rate. So the MYC would be about 40 to 60%, and the Dlk1 would be about 30 to 40%, and it was always lower for each cohort. But when you look at 6 months, the Dlk1 has actually caught up with MYC, and when you actually take that initial transduction rate into account by dividing the GFP positive proportion by the initial rate, actually the cDLK1 vector containing cells seem to have an advantage compared to the MYC containing cells in terms of the reconstituting the recipient mice. And when you look at the mDLK1 vector, the difference is actually a bit more in that even without taking into account the initial rate of transduction, you can see it's actually still is significantly higher for the mDLK1 group versus the MYC group. So and also the difference here is bigger as well. So the mDLK1 vector containing cells also seem to have a competitive advantage compared to the MYC containing cells. So in summary of my second chapter, Dlk1 overexpression can be readily detected by PCR and Western blot in human and mouse cancer cell lines. And I've shown that the Dlk1 positive cells sorted from the 2 human leukemia cell lines do not show any difference in clonogenicity. And the overexpression vector works and leads to sustained high level of Dlk1 expression in hematopoietic cells at long term. And Dlk1 overexpression does not affect peripheral blood count and does not cause any significant differences in survival or in acute leukemia or in GFP-positive leukemia. But the cDLK1 and mDLK1 containing cells seem to have a competitive repopulation advantage compared to the MYC-containing cells. So I'll move on to the next section, which is my last section. And we've decided to study Dlk1 in angiogenesis because of its link to the Notch signaling pathway. And in this paper published in 2012, the absence of Dlk1 was shown to be correlated with abnormality in postnatal mice retina, and this was thought to be due to Notch pathway dysregulation in the endothelium. So my plans were to study the developing blood vessels in the retina and other organs in the postnatal mice using Dlk1 EGFP mice that I mentioned before, and also look at the developing blood vessels in the retina of the mice that lack Dlk1 expression or conditionally lack Dlk1 expression in the endothelium. And in the end, we just went with the latter, the conditional lack, because as I've said before, the knockout mice are very difficult to get, so we've decided to create the conditionally knocked out mice to study the retina. So the first one, for the first objective, we looked at postnatal day 5 mice organs and looked at the endothelium of those organs and found that on day 5, Dlk1 is significantly expressed in the lung and muscle endothelium. So the top panel here is GFP mice and the bottom is wild type. And heart, kidney, lung, and muscle endothelial cells by the FITC fluorescence. So in heart and kidney endothelial cells, there's no difference between the GFP and wild type mice in terms of the FITC fluorescence. But when you look at the lung and muscle, there is significant shift to FITC positivity in the GFP mice and also in the muscle compared to the wild type where there's none. The blood vessel, however, is not just about the endothelium. There's other important cell types there as well. So endothelium is supported by another mural cell type called Parasite, and this is all wrapped up in a sleeve by a basement membrane. And I've decided to stain the retina for endothelium, which is by PKM1 antibody, Parasite using Ang2 antibody, and Dlk1 using the GFP antibody, because I'm using the Dlk1 GFP mice. And when you look at these images, it's hard to tell whether Dlk1 is expressed in the endothelium, which was what was claimed before, or whether it's expressed in parasites. It's difficult to tell. But when you look at high power, this is endothelium and parasite and the Dlk1 in blue. When you look at the endothelium versus Dlk1, the blue cells seem to be kind of lying alongside it or wrapping around the endothelial cells, whereas if you look at the parasites, they seem to actually co-localize together, which makes it so with the combination of red and blue, it actually turns into purple. So it seems to be the parasite that expresses Dlk1 and not the endothelium. So to prove or to really look at what endothelial conditional Dlk1 knockout does in the postnatal retina, we've created this Ty2-Cre endothelial conditional knockout mice by having a homozygous flox Dlk1 mouse mated to a Ty2-Cre transgenic mice. So the Ty2 is expressed in all endothelial cells and most of the hematopoietic cells. So what you get at the end is 50% experimental mice where the Dlk1 is deleted in all endothelial cells and in most of the hematopoietic cells, and 50% control mice. So using the conditional knockout mice, in the transgenic mice versus wild-type mice, on gross examination, there's no significant abnormality, but more detailed statistical analysis on these are pending. So just a quick summary on the part 3. Dlk1 is significantly expressed in the lung and muscle endothelium of postnatal pups. And the retina, it's expressed by the parasites and not the endothelium of the developing blood vessels. And when Dlk1 is conditionally knocked out, no gross morphological differences can be observed. So, so we've come to the end of my talk, which is the key points of my PhD project. So Dlk1 is an imprinted gene. It's a member of paternally expressed genes in the very important Dlk1 DO3 imprinted region, which is gathering a lot of interest recently as an important player in stem cells and tumor biology. And I think my in-depth study has contributed to the current scientific knowledge in this area. It's expressed at high level in hematopoietic stem cells of the fetal livers, but the expression decreases with age. The Dlk1 knockout leads to increased clonogenicity in both fetal liver and adult bone marrow, and to significant defect in the reconstitutive ability of short-term hematopoietic stem cells in the adult bone marrow. On the other hand, when you overexpress it, it seems to confer competitive advantage in reconstitution, which might contribute to leukaemiogenesis, but by itself, it doesn't cause acute leukemia. So the Dlk1 is expressed in parasites, as I've shown you, and not in the endothelium. And it's highly expressed in the endothelium of postnatal lung and muscle tissue. And I mean, this is just what I'm thinking, that because Dlk1 is so commonly expressed in human leukemia, it may be a useful adjunctive target in treatment against acute leukemia. And I think there might be a potential for Dlk1 to be used to expand hematopoietic stem cells, not only because it seems to confer some competitive advantage, but also there's been publications in the past and recently showing that activated Dlk1 expression seems to be important in maintaining pluripotency and using soluble Dlk1 to expand cobblestone areas. So I think with all those, there may be a potential for Dlk1 to be used to expand stem cells. So that's the end of my talk, and I'd like to say a very big thank you to my supervisors who are sitting there. And thank you very much for your support through very difficult periods in my life where I had a baby in my second year. Very, very grateful to my PhD committee as well. And everybody in cancer and hematology who really had to show me from absolute basic about everything, because I really didn't know what to do with anything, from pipetting to just about everything. I had to learn from these people, and I'm very grateful. Thank you. Any questions will be greatly appreciated. Yes.
In the fetal liver, it appeared to me there was one isoform. Is that the isoform expressed, or is just
I did notice that in some tissues, some isoforms, all isoforms are expressed and some don't. And yeah, as you said, in fetal liver, the ones that seem to be expressed seem to be the long one and the shortest one. I don't know. I don't know why, but it does seem to have both of the complete and the membrane isoforms. And it does seem to, in different tissues, it does have different, seem to have different pattern of isoforms. The Dlk1 antibody actually detects all of the isoforms, yeah.
Mani.
Nice talk. So I wonder, have you thought about the difference between your fetal liver transplants and your adult bone marrow transplants? So, because normally I think when you transplant fetal liver stem cells, they essentially turn into adult stem cells anyway, particularly in serial transplants. So have you considered taking your imprinted knockout and doing fetal liver transplants with that model to see what happens in that case, whether it's a genetic difference rather than the stem cell difference?
Yeah, the fetal liver transplants, I actually thought that doing the fetal liver transplant is going to show a bigger difference.
Yeah.
Because, as I've said, the adult imprinted knockout is undetectable. The wild type has got a little bit of expression, but the fetal liver, the Dlk1 expression is quite high. So I thought if I took the knockout versus the wild type fetal liver, the difference is going to be huge. But that actually didn't turn out to be the case. I can think, been thinking about why that might be. I mean, my theories are maybe it's because the role of Dlk1 in fetal liver is that maybe the actions are different in fetal liver versus in the adult bone marrow. Or because the fetal liver hematopoietic stem cells are intrinsically, when they're first used, they are different. They're more proliferative. They compete against bone marrow and they win every time. And maybe just that huge, much more proliferative nature perhaps kind of compensates for the knockout of Dlk1. So those are the things that I thought might be why I don't see a difference in the fetal liver. But I was quite surprised by that.
My thoughts were that when you've got secondary and tertiary and quaternary transplants, they're no longer fetal liver cells anymore.
Yeah, that's true.
So why would they behave differently?
Yeah, that's a good point. Yeah, I'm not sure about that actually.
I think the most impact was in the first transplant, and whether or not there are fewer functional stem cell units in the first recipient was probably the answer. And I guess the question about the using imprinted versus knockout, we don't know necessarily, has been shown in other tissues how imprinted it is throughout development. So to answer the question definitively, the knockouts were used from those fetal liver experiments.
So in the retina you saw that Dlk1 was expressed in the parasites rather than the endothelium. When you looked back, some tissues had Dlk1 expression and some didn't. Why do you think that is?
Yeah, I don't know the answer to that. I think it's just one of those, that Dlk1 is sometimes expressed in some cell types in endothelium. And in the brain, though, I know from previous publications that it is mainly expressed in the parasites, in the CNS. So it's probably not surprising that in the retina, it's not the endothelium, but the parasites, because in the rest of the brain, the Dlk1 is expressed in the parasites. So yeah, I don't know why in some organs it's expressed in the endothelium, but in the brain, and including the retina, it's expressed in the parasite. It's probably got different roles in different tissues.
Sophie, you should mention the phenotype of your Dlk1 knockout lung.
Yeah, so when we knock out Dlk1 in, I'm talking about imprinted knockout, because obviously the homozygous knockouts will die. The imprinted knockout mice, the lungs are very abnormal, probably because of the fact that it's highly expressed in the lung endothelium. So the blood vessels are very abnormal, and even the alveolar cell structures are very abnormal. So it does seem to have a significant impact in the lung formation.
Time for one question. If nothing there, it remains for me to thank Sophie, who's really been an exemplar of what a PhD thesis is about. Well-conducted experiments, well-designed experiments, and conducted with rigorous scientific rigour, really. So thank you very much.
Thank you.